This is the lecture about the action potential and the parts of a neuron. This material is usually the most difficult for students, so if you have trouble, please make sure that you read your book, review the lecture multiple times, and ask me questions either on the discussion board, via email, or stop by my office. I am definitely happy to help. My background is neuroscience, and so this is my favorite stuff. So the first thing we're going to cover are the parts of the neuron. You do need to know about the neuron and you need to know its main parts. The first main part is actually we're going to start with the center of the cell, which is the circular structure. It kind of looks like a little eyeball in the very, very center. And that's what's called the cell body or the soma. This is just like any other cell that you've probably studied in a regular biology class. It has a nucleus, it has DNA, it has mitochondria, it has all of that kind of stuff. Now, in a psychology class, we're not interested in that kind of stuff. What we're going to try and do in here is focus in on the parts that make a neuron special. And those include the dendrites and the axon. Now, the dendrites look like tree branches coming out of the soma. And the dendrites are what receives the signal. So when a cell first gets a signal, it starts in the dendrites. The axon is the kind of funky looking thing that's at the bottom of the cell in this diagram. It kind of looks like what I like to call a sausage link. We'll talk about why it looks like a sausage link later in this lecture. In terms of what the axon does, it sends the signal to the next cell. So imagine if you had one neuron with an axon and it was connected to the next neuron where there's a dendrite. And then the axon of that neuron connected to a new neuron with its dendrites. Some people find it helpful to think of the dendrites and the axons as like the pitcher and the catcher. So the axon would be the pitcher and the dendrite would be the catcher. Now our next question is going to be, well, what is the ball? What is the signal itself? Well, there are different kinds of signals in the brain and nervous system. The first main kind of signal that we're going to talk about is an electrical signal. And the main electrical signal is called the action potential. This is the electrical signal inside of a neuron. Now the action potential looks something like the graph that's on the slide. The voltage is on the vertical axis and time is on the horizontal axis. In terms of volts, we actually do have electricity running through our bodies, but we call the electricity in our bodies ions. And specifically, we have sodium and potassium ions in our bodies. So I want you guys to try and understand what's going on with this graph. If you start on the left side of the graph, notice that the red line starts off flat. This is a neuron that has nothing going on, so there's no activity. And then notice where it says stimulus and there's a little arrow. Notice that the red line starts to go up. That means that that graph is getting more positive. The cell, the charge of the cell is getting more positive. How does that happen? Well, sodium is actually entering into the neuron during that time. Remember that sodium is charged positive. And so when positive goes inside the cell, that is making the cell more positive. That's why that line goes up. Now, once you get to the very top of where the red line goes, and then it starts to kind of come back down, that is when potassium decides to leave the neuron. Now remember, potassium is also charged positive, but potassium is leaving, so the cell is getting less positive, and that's why the voltage goes down. So for now, you need to understand that the first thing that happens with the action potential is that sodium enters the neuron, and that makes the neuron positive, and therefore our line goes up. After that, potassium exits the neuron. Potassium is also charged positive, so when potassium leaves, the cell gets less positive, and that's why my line goes down. The next thing I want you to look at closely is the things that are circled with the little blue circle, these little blips. And if you look, you can see they are labeled as failed initiations. If you think about when you walk into a room and let's say someone hasn't taken the garbage out lately, it might smell when you first walk into the room. And initially, imagine that that signal gets all the way up and you get a whole action potential through when that signal first comes in. But after a while you're in there, your brain, your neurons, your nose, and so on eventually say, well, this is nothing new. I've been around this smell for a long time. And so that signal stops getting sent all the way through. They're kind of like failed initiations. This also happens with the auditory system. So for example, you walk into a room or with a fan on, or someone turns on a fan, and it's very noticeable. But after a while, you stop noticing it. You stop paying attention to it, and probably don't even realize that there's a fan blowing in the room. Again, the first time you hear it, that signal is making it through. It's as if the sodium came up and the potassium comes down and out, and you get a full action potential. But after a while, you stop getting a full signal, and that would be like the failed initiations. Now the other kind of key term that goes with failed initiations is this term, threshold. You might ask, well, when do I get a failed initiation and when do I actually get a signal, an action potential? Where's the cutoff? Well, the cutoff is the threshold. Again, the key is that not all incoming signals are strong enough to create an action potential. And to be strong enough, they have to reach the line at threshold. Now, along with this threshold idea, right, is another kind of really key theory about neurons and neuroscience and action potentials. Now, if your incoming signal does reach threshold, you do get a full action potential. If you don't quite make it to threshold, in fact, nothing happens. It's a failed initiation. The interesting thing about this is there is no middle ground. There's not like, well, what happens if you almost get there? Do you get a small action potential? Or what happens if you go way past threshold? Do you get a really big action potential? In neurons, you don't get small action potentials. You don't get big action potentials. You either get them or you don't. And the term that goes along with this is an all or none response. You either get it all or you don't get any. People like to equate this to like a toilet, right? When you flush a toilet, you either, flush the whole way or you don't flush anything down, right? It's either there or it's not. Sometimes people like to talk about guns this way, right? You can't really squeeze the trigger and like shoot the bullet a little slower, right? You either squeeze the trigger and get a shot or you don't and you get none. Maybe you didn't squeeze quite enough. You didn't reach threshold. One more thing to look at with this graph is that little blip at the very end that's circled here. It is what is called the refractory period. The refractory period is a time when the neuron cannot be active. It's basically like a rest period. The cell is kind of resetting. If you go back to that toilet idea from the last slide, once you flush the toilet, you have to wait for it to refill before you can flush it again. Well, essentially in a neuron, once things have started to go, you got to wait for things to reset before you can have another signal again. Now remember, if you look at the graph, the line went up at the very beginning, but why did that line go up? Well, it went up because sodium came inside the cell. That must have meant that sodium started off outside. Now the line goes down because potassium leaves the cell. Well, potassium then must have started off in the cell. So now we're down in that bottom, in that refractory period, and sodium is inside and potassium is outside. Well, that's the opposite of where they started, right? Sodium should start out and potassium should start in. But instead we have sodium in and potassium out. So during the refractory period, they essentially must switch back and forth before that cell can become active again, before more sodium can come inside the cell and start things up for a new signal. There are 3 more parts to know for the neuron. These are 3 smaller parts, and they are all part of the axon. Remember that sausage-like thing. They are important because this is where it all happens. So this is really going to help you understand what's going on in a neuron and how the action potential works. The first part to know is called the axon hillock. And the axon hillock is the connection between the cell body and the axon. It's very important because this is where the action potential begins. This is where the electrical signal starts. And this is where threshold gets measured. So this is where you go, okay, well, was there enough signal? If so, give me an action potential. If not, nothing's going to happen. Now let's talk a little bit about why the axon looks like a sausage link. You may notice that there are these kind of bluish, whitish structures that appear to be kind of surrounding the axon. That is what's called a myelin or myelin sheath. Now imagine you have a wire miles long and you try and have an electrical signal go through that wire. By the time you get to the end of that wire, most of that signal will be gone. How do we make that signal make it to the end? Well, wrap something around it. You do what's called insulating it. That's essentially what the myelin is. Myelin is actually composed of fat, and fat is a great insulator. It will keep that electrical signal strong, that action potential strong. Another way to put it is, let's say I have a hose, like a 10-mile long hose that goes from Rio Hondo and goes down into Uptown Whittier. Let's say I start with boiling hot water at Rio Hondo. Will I end up with boiling hot water by the end? 5-10 miles later? Probably not. And what has happened? Why has the water cooled off? Think about it, right? The heat has probably left. The heat has dissipated. So how could I fix this? Let's wrap that hose in something, some sort of insulation that will keep the heat in. Again, that's what myelin does. Myelin is basically something wrapped around your hose to keep your signal in. In this case, it's not heat, it's electrical, but it's the same idea. Now, there is one issue with this from an engineering standpoint, and that is if you look at all of the myelin, there are these breaks that are there. And those breaks are there for a reason, and they have a special name. They are known as the nodes of Ranvier. In theory, this is a very bad thing, right? Because the insulation is going to keep our signal strong, but once you get in between insulation, your signal's just going to get lost very quickly. Well, the nodes of Ranvier are basically where your signal starts over. Why does this happen? Why does it get regenerated? Why does it need to start over? Well, there is no such thing as a perfect insulator. So if the entire axon was wrapped in myelin or some other form of insulation, your signal still might not make it to the end. We do have some axons that are as long as a couple feet in our body. So this could be a very long path that your action potential would have to make it down your axon. So what happens is your signal starts at your axon hillock, and then it starts to move down the axon. The electrical signal moves down, and as it moves, it does lose a little bit of its signal through the myelin. There is no perfect insulation, so it will get a little smaller. Not too small, but a little smaller. But then it gets to that break, that node of Ranvier. And when it gets there, it gets basically like restarted, regenerated. It starts over again. It gets its original size. And then it moves down the next bit of myelin. And when it moves down that next bit of myelin, it gets a little smaller again. But it gets to the next node, to the next break, and whoop, it comes back up to full size. And this is essentially how it comes all the way down the axon, getting regenerated over and over and over and over and over again, the whole way down. If I blow up the picture to give you a slightly better idea of what's going on, you can see that at every node of Ranvier, or at every break, the signal is getting regenerated, and that it travels down the axon in that way. This will allow the signal to stay strong as it goes. Again, I know these are very difficult concepts, and if you do have questions, please let me know. This right here, the action potential and how it works, is the most difficult part of probably the entire course. Now I put this slide on, and it says not all neurons look alike, and I do this just so you know that everything I give you in here is kind of a simplified version of what's going on. And sometimes students, they leave the course and they go, oh, well, that's what a neuron is. And I just want you to see that even what I've been showing you, well, that's one kind of neuron. But there's a whole lot of other kinds of neurons. So you can see that probably the closest thing that what we've been talking about is the multipolar neuron here. But you can see that there are bipolar neurons, unipolar neurons, really funky things called pyramidal cells. Really across the top you can see there are dendrites on each one. And then along the bottom you can see that each one does have axons. So all neurons do have those things, but they do come in different shapes and sizes, and they all do slightly different things. Okay, so what happens when we get to the end of the axon? Remember, one neuron's axon, the pitcher, sends the signal to the dendrites of another neuron, to the catcher. The place where the axon and the dendrite meet is called a synapse, and this is where communication between neurons occurs. And the signal that occurs here is not electrical anymore. This signal is the other kind of signal we see in neurons, which are chemical signals. And the chemicals that are used in neurons are called neurotransmitters. So what exactly does go on here? Well, when the action potential is coming down, it causes all of these little packets of neurotransmitters. You can see these little circles in that picture with little dots in them. And it causes all of those to leave the axon and to go over to the dendrite. This can then start a new action potential. So the cycle is that you get this action potential that starts, moves its way down the axon, neurotransmitters get released, and those are chemicals, and the chemicals then go over to a dendrite, which then picks up the signal, and the dendrites then can start a new action potential, an electrical signal, which goes through the axon, and then at the end of that axon, more neurotransmitters get released, more chemicals, and so our signal is actually going back and forth between electrical action potentials and chemical neurotransmitters. Think about it goes action potential, then neurotransmitter, action potential, neurotransmitter, action potential, neurotransmitter, or electrical, chemical, electrical, chemical, electrical, chemical. Now, what do we mean when we say neurotransmitter? I'm not going to go into this in too much detail. You don't need to know it in too much detail, but there are a few big neurotransmitters that I'd like you to at least be familiar with. The first is what is called acetylcholine. Now, acetylcholine is the neurotransmitter that links your nerves to your muscles. The reason you can move is because of acetylcholine. If you flex your arm or move your hand or talk or anything along those lines, that's because acetylcholine is being released at those synapses. It's also been implicated in Alzheimer's disease, so it doesn't do just the one thing, it seems to do a few other things, like memory, like Alzheimer's. Now, what happens to an animal when it's bitten by a cobra and gets venom? Notice the picture on the right. It does get paralyzed. Well, how would that work? Remember that acetylcholine is making us move. So if you can block the acetylcholine, you would essentially paralyze an animal. The second neurotransmitter is called dopamine. Dopamine is extremely important, and you might have even heard about it before outside of this class. It seems to be involved in learning, emotions, schizophrenia. When you talk about drugs, which we'll talk about later in the class, it's very strongly involved in addiction. When dopamine is released, it feels very pleasurable. So this seems to be why people are likely to get addicted to drugs, because the drugs make it feel good. The third and other neurotransmitter that you might have heard of is what's called serotonin. Serotonin is kind of famously linked to depression. If you don't have enough serotonin, you are likely to be depressed. Antidepressant drugs like Prozac, Zoloft, Lexapro, these all work by giving you more serotonin. This marks the end of this lecture. If you do have any questions at all, please let me know.